for two reasons. One is that hopefully most, 95, 99% of that is a review. But also, I'm going to go back to the various points again and talk about the various organelles and functions in more details. So that's why I skim through them quickly. Okay? We are talking about cytoskeletons, and we are actually, so just I'll pull back a couple of slides. We're talking about the three different types of cytoskeletons, right? Microfilaments, microtubule, and intermediate filaments. Microfilaments and microtubules are only found in the cytoplasm, not cytosol, but cytoplasm. And intermediate filaments are found in both the cytoplasm as well as the nucleoplasm. So in the nucleoplasm, to distinguish intermediate filaments from cytoplasmic intermediate filaments, I will be calling the nuclear intermediate filaments the nuclear scaffold. Okay, so I will call it the nuclear scaffold. Sometimes later on, there's yet another name. So in science, there are multiple names for a lot of things. To remind you, so this is where we left off, and let's pick up from here. So we're going to concentrate on microfilaments. And what I'm going to, just to let you know how it's going to set up, I'm going to talk about each one of all three cytoskeletons in terms of structure, and how assembly of the structures are, what are they, where are they found, and then later on come back and talk about them in more details. Okay, so I layered the talk so that I don't hit you with everything first and then you forget everything about microfilament when I talk about microtubules. Okay, the reason why I do this is because a lot of the structures as well as functions parallel for these three cytoskeletons. They have similarities. So I want to show you the similarities so that when we go into the details, the regulation of assembly and disassembly and thereby regulation of function, that will, you know a lot of the details, and the regulation would be the differences. Okay? So anyway, so microfilaments. Microfilaments, the subunit is a G-actin, like I told you. Most of this, don't worry too much about this slide. It's just a prompt for me to tell you that the monomer for microfilament is a G-actin. Okay? This picture is just a pretty picture for now. Don't try to memorize it, but basically what you have is you have each of these circles represent a G-actin, or an actin molecule. They're linked end to end, and then if you take an end to end to form what is called a protofilament. Okay? And then if you take two of these protofilaments and you wrap them up and twist them into almost like a double helix, a DNA double helix, then that right there is a microfilament. So don't try to memorize everything of what I just said. The reason why I say that is to give you an idea of the complexity, right? It's not a matter of linking monomers together. It's linking monomers together, and then you have two of these, and then they twist to form an actual microfilament. Okay? So this is polymer assembly. So this is the assembly of microfilament. Is a little more complex again than just linking the monomers together. Don't have to remember the structure or anything. The other thing, however, to remember is that the linkage of the G-actin requires ATP. Okay, so here's the deal, and I'll repeat this later on, but let me introduce it now. G-actin that are bound by ATP, already has ATP bound, can be used for polymer assembly. However, if a G-actin is bound by an ADP, then that G-actin cannot be used. Don't say that in one form is an active form and the other one is in inactive form. This has nothing to do with a function. This has to do with binding, right? So a GTP that has an ATP bound to it would be in the proper, would have the proper conformation or structure to be used for assembly. Whereas a G-actin that has an ADP bound to it would not have the conformation, would not be in the right conformation to be used for polymerization or polymer assembly. Okay? So those are the two things that I want you to get out of this slide. One is that it's more complex than just linking the G-actin together. The second is that in order for a G-actin to be used to be linked together, that G-actin must be bound to an ATP. And if it's bound to an ADP, then it won't be used. Some of you might ask, well, what about G-actin that is not bound to either ATP or ATP? That doesn't happen. G-actin needs to be bound to either an ATP or a GDP. So the addition of the ADP or GTP, what is that? That's translational modification, right? So it has to have translational modification. Where and what kind of functions, and this looks like a lot. And by the way, we're going to go into details about each one of these things. So this right here is a summary of what we're about to talk when we talk about microfilament. Okay? Don't let it scare you. I will try to talk about each one in details and why they look the way they do. And so, if you were to think of that red line right there as a microfilament, one microfilament, meaning that you have two protofilaments wrapping together, right? So that's a microfilament. And if you take a bunch of microfilaments, they can be bundled or aggregated in different ways. There are three different ways that they can be bundled or aggregated. They can be loosely bundled to form a contractile bundle of microfilaments. So this is a loose bundling. So, and the idea is that there's space in between to actually jam more stuff in it. Okay? And we'll talk about what the other stuff might be. There is a type of aggregation or bundling where it forms a gel-like network where the microfilaments are not bundled like if you take a bunch of twigs and you don't bundle up into it like that. They just form they form angles to one another. Okay? And that's a gel-like structure. Or the third type of bundling or aggregation is by tight parallel bundling, where very similar to the contractile bundle, these microfilaments are in parallel, but they're tightly bound so that there's nothing that can come in between the microfilaments. So these are the three types of aggregation or bundling that can occur in microfilament. Microfilaments will have to be bundled in one of these three types. So a microfilament will not just stand there by itself, right? Microfilament that is like that will be too floppy. Okay, and we'll talk about the skeletal muscle very specifically later on. In reference to these three types of bundling or aggregation of microfilaments, the contractile bundle of microfilaments are found actually in the center bottom. So let me. So those three types of bundling, right? So let me go back to the very first slide that we looked at, and it's actually more apparent. Okay, so the contractile bundling is found right there. The stress fibers, remember I said that this cell is sitting on a plate of some kind, right? And that's where the stress fibers are. Okay, so if a cell is not attaching to a bottom of some kind, even in your body, if it's just floating around, you would not have stress fibers. So therefore, you will not have contractile bundling. So contractile bundling are special, and they're only formed when needed. And one of the places that's needed is within the stress fiber right here. Okay, so that's where contractile bundling is. Within the cell cortex, the type of bundling is the gel-like network. And then remember, I also point out that every now and then you have these later on we'll find out that they're called filopodia or microvilli, that you also have tight bundling that come out of the cell cortex. Okay? So that's where, so the type of aggregation of bundling of the microfilaments depends on where the microfilaments are. And depending on how they are bundled and where they are, that's how they perform their function. Does that make sense? So it's not just whether it's microfilaments, it's how they are bundled. It's not just how they are bundled, it's where they're found. Okay? Because they won't perform their function unless they're found in the proper place. So that's that. And I just mentioned microvilli. So this is an example of microvilli right here, and you would have tight bundling in the microvilli. For instance, small intestine, right? The lining of your small intestine, these cells would have these finger-like structures called microvilli that extend out. Right? During cytokinesis of cell division, if you remember contractile ring, right, that is formed through the microfilament assembly, and the type of bundling here is actually the contractile bundling. And later on we'll talk about why it has to be a contractile bundling and how the cell is squeezed in the middle. And then lastly, you probably, since first grade, you heard about the sarcomere and how muscle contract, and the thin filament of the sarcomere is made up of, or are made up of microfilaments, or better known as actin filaments. And then the green, the thick filament is made up of myosin filaments. And so to give you the punchline, the reason why the contractile bundle needs to have space is because that is where the space is where the myosin filaments, or the thick filaments, will get added. Okay, for the purpose of contraction. So whenever you see a contractile bundle, you know that the cell can perform contraction. Not just in the form of a sarcomere, but also in the form of, like here, cytokinesis requires contraction. Okay? So structure function. If you have a certain structure, you have to have that function. We'll talk about why the gel-like structure of the microfilament occurs in the cell cortex later. Polarity. So a little bit more about G-actin as well as microfilament is that they're both polar. The G-actin is polar, and meaning that it has two distinct ends, and the microfilament is polar. It has two distinct ends. One end is called the minus end, and the other end is called the plus end. Okay? So later on we'll talk about the plus end and the minus end of a microfilament. We also will talk about associated proteins. So a microfilament, its function, in order for the microfilament to function, to have a function, you can't just have G-actin linking and then forming a microfilament. There have to be non-G-actin, or proteins that are not G-actin, associating with the microfilaments, and these are called microfilament associated proteins. And one of which I already mentioned, a molecular motor or motor protein is a microfilament associated protein. And so the microfilament associated proteins help the microfilament perform its function. Depending on the type of bundling or aggregation, as well as depending on where the microfilaments are found, then different microfilament associated proteins will be attached to the microfilament. Does that make sense? So they're specific. Very briefly, in general, the assembly and disassembly of microfilament is, for the most part, assembly and disassembly would occur, let's say you're looking at this, would occur toward the plus end. So if we say that that's a plus end, and that's the minus end right here. Okay? assembly and disassembly of the microfilament again occurs mainly at the plus end. That means that it has to start from the minus end, right? It goes from the minus end toward the plus end. Very much so like when we talk about, let's say, DNA, nucleic acid synthesis. It goes from the 5' end toward the 3' end. So nucleotides are added to the 3' end, right? In this case, G-actins that are bound to ATP are added to the plus end. And however, also removal of disassembly may also, occurs mainly at the plus end as well. But it's a little bit more complicated than that. Okay? The idea is that microfilaments are very dynamic. They do one of these. So if this, my hand represents where the minus end of the microfilament is, then the plus end would do one of these things where it fluctuates like this, right? It's very dynamic. You have assembly and disassembly constantly. As you're sitting there right now in your cells, you have microfilaments being assembled and disassembled continuously, okay, in large amounts. And so all of that has to be regulated. So that's an introduction to microfilaments. Let's stop there. Okay? Let's talk about microtubules, and a lot of assembly and disassembly of microtubules also are parallel with microfilaments. Okay? For microtubules, the monomers are tubulin. Tubulin is actually there's a family of tubulin, alpha, beta, and gamma tubulin. And really, only the alpha and beta tubulins will be used as monomer for microtubules. The gamma tubulin is used for something else. Okay. So that for now, let's hold off on that. We'll come back to the MTOC later. Okay, so for microtubules, just like the picture for microfilament, I don't want you to memorize it. There are two things, again, that I want you to get out of this picture. One is that the assembly of microtubule is even more complex than that of microfilament. In fact, you have to have not just 2 protofilaments, but you have to have 13 protofilaments in most cases, and sometimes only 10. So it's variable. You have 13 of these protofilaments lined up side by side, and then they form a cylinder. So they don't twist, they form a cylinder, right? And that cylindrical shape containing 13 protofilaments get longer and longer and longer, and that is what is referred to as a microtubule. And then microtubules are also aggregated, sometimes, not always, but sometimes aggregated to form other structures. Okay? But for now, do realize that the assembly of microtubule is more complex. Since the assembly is more complex, the disassembly is also slightly more complex as well. Okay? So that's one thing that you need to get out of this slide. The other is that, as opposed to G-actin requiring an ATP bound to be used for assembly, tubulins require a GT bound, a GTP bound to it to be used for assembly. But the same thing occurs where tubulins that have a GTP bound can be used for assembly, and then tubulins that have a GDP bound cannot be used for assembly. Okay? And the same question is, what happens to tubulins that don't have either GTP or GDP bound, but that does not happen? Okay? Yes, ma'am.
So, as far as back to the active metals, if there's none of those without an ATP or GDP.
Can you say that again?
You said if there are none without GDP bound for the actin, there are none without ATP or ATP bound.
For G-actin.
Yeah.
Right. That's right. Yes, sir.
So the tubulin heterodimer is the monomer.
Right. So again, that's the other complexity that I didn't want. So instead of G-actin being the monomer and you link the monomer, tubulin actually, the alpha and beta form dimers first, and it's the dimers that get linked. same difference really, but we're not going to go into that much detail. But yes, you got it. You got it. Okay. So only those 2 points. Well, since we have it here, microtubules and tubulins are also polar. So therefore, a microtubule also similar parallel with microfilaments has a plus N versus a minus N, and also the assembly and disassembly for the most part, the majority of the time occurs at the plus N. And we'll learn very quickly why the minus N for both types of cytoskeleton are more stable and don't enter into assembly and disassembly. But again, so microtubules also has a plus N and minus N, and assembly and disassembly occurs mainly, not 100% of the time, but mainly at the plus N. Okay. In a cell, if you have these a test tube with G-actin, or you have a test tube with tubulin dimers, then both ends will be involved in assembly and disassembly. So what I just said applies to an in vivo condition. Okay. Growing, shrinking, even pretty pictures right here. So where are microtubules found, and what are some of the functions? Microtubules can, as I showed you, can radiate out from the nucleus. So the nucleus is somewhere right here. The cell is a big old cell, and microtubules actually radiate outward. And you saw that picture from the very first picture, right? Microtubules can also be found and function during cell division in the form of spindle fibers. Okay? So there are 3 types of spindle fiber. Here's the kinetochore spindle fibers. There are some of them that don't connect to the chromosome, which are called polar spindle fibers, and then you have the aster spindle fiber. But they're all microtubules, right? They're all spindle fibers. Let's see. In terms of other places that and functions that where microtubules are found in axons. Axons in neurons are created from the assembly of microtubules. So the axon has a plasma membrane out here, and underneath is the microtubule. Dendrites, also of neurons. Okay. In cells that have flagella and cilia, not microvilli, are formed from assembly of microfilaments. Cilia and flagella are formed from the assembly of microtubules. Okay. Cilia looks very much like microvilli. But since they are formed from different types of cytoskeletons, they have different functions. So in this case, 2 things that have similar functions but have different functions. That they're called analogs, right? So analog is whenever you have similarity in functions, but different similarity in structure but different functions. And homologs are similarity in function, and most of the time also means similarity in structure. Okay, so we'll talk about flagella and cilia. In plant cells, you also have and everything that we've talked about so far applies to an animal cell. Plant cells also have cytoskeleton. But they don't have intermediate filaments. So plant cells have microtubules, have microfilaments, but do not have intermediate filaments. Believe it or not, prokaryotic cells also have cytoskeleton. Okay, and we'll touch on that briefly later. So far, so good. So plus N minus N. Let me flip forward a couple of times. Okay. Okay. So we'll talk about all of this in a minute. Microtubules are also dynamic. So they do one of these things: assembly, disassembly. Okay. very similar to microfilament. So you see that microtubules and microfilaments are very much similar, okay? Intermediate filaments are completely different. Number one, intermediate filament, there isn't just one monomer for intermediate filaments. Monomers for intermediate filaments are cell-specific, okay? Depending on the cell type, then you would have a certain intermediate monomer for intermediate filaments, and then you have intermediate filaments. So that's one difference. Another major difference between intermediate filaments and both microfilaments and microtubules is that intermediate filaments are very, very stable. They're not dynamic. That's the second difference. So they're very stable. And the third difference is the fact that intermediate filaments are not polar. The two ends look the same. Right? The two ends are not different. Okay. And like it's mentioned here, plants and single-celled organisms, mainly prokaryotic cells, but some protists as well, do not have intermediate filaments. Because intermediate filaments, because it's so stable, it provides the cell structure, right, and support for the cell structure. Well, single-celled organisms change their structure. The shape of the single-cell organisms are more amorphous, so they have to change too much, right? So they don't have intermediate filaments. Intermediate filaments, if you look at, if you learn, and we did learn about squamous versus cuboidal cells versus columnar cells, right? The reason why a columnar cell is columnar is because of the structure is provided by intermediate filaments. The reason why a squamous cell is squamous is because intermediate filaments provides that structure. Okay? And in order to go from one structure to the other, the intermediate filaments has to be rearranged, and that takes a long time, is because, again, intermediate filaments are not dynamic. So it takes almost practically, well, not forever, but a long time for intermediate filaments to change. Okay? So don't memorize this. This is just to tell you that as complex as microfilament assembly is, microtubule assembly is more complex as microtubule assembly is. Assembly of intermediate filaments are even more complex. Okay. So we talked about the difference between nuclear support versus cytoplasmic support. Right? So intermediate filaments in the cytoplasm will provide not only the shape of the cell, but support for the cell. Okay, so the support for that shape. Intermediate filaments in the nucleus also not only provides the shape of the nucleus, which is kind of roundish, but also the support for it. Right? And if you've ever taken histology or sometime in, I think in anatomy, in Bio 22, you learn a little bit of histology also. You find that some cells have a big round nucleus, and some cells has a flattened nucleus, right? So the shape of the nucleus can be different among different cell types, and they are again created and supported by the nuclear scaffold. Okay. Let's see. So, in addition to that, how else are intermediate filaments involved with cell function? Intermediate filaments, as well as microfilaments, but not microtubules. So here's a similarity between intermediate filaments and microfilaments, is that they provide an anchorage. They provide a stable anchor for cell adhesion. So whenever a cell interacts with another cell, or a cell interacts with the extracellular matrix, right, they do that through the proteins on the non-cytosolic side of the protein, of the plasma membrane, But on the cytosolic side, right, these proteins must be anchored on somehow as well. And again, we'll talk about the details of that later on. So then intermediate filaments as well as microfilaments would provide anchorage for cell adhesion. We already talked about lamin, so the nuclear scaffold is actually created by assembly of the monomer called lamin. So lamin is the monomer that is used to create this nuclear scaffold. And all nuclear scaffold for animal cells that we know of contain lamin. Sometimes the nuclear scaffold is referred to as the lamin filament. Okay. Where else do you find intermediate filaments? Skeletal muscle. So if you have the, let's say here's a sarcomere, and anybody not know what a sarcomere is? It's okay if you don't know. Okay. So roughly, muscle contraction, the cells within, like for instance, especially skeletal muscle and cardiac muscle, they contract. Well, there are individual units within each, within the cardiac muscle cell and the skeletal muscle cells called cardiac muscles, and they contain a Z disc where the microfilament attached to. Okay, and then they are formed in they are aggregated in a more contractile bundle style. And then in the middle you have myosin. So this is a contractile unit of striated muscle. They're called striated muscle is because everything is lined up parallel like this. So if you look under the microscope, you see lines. Okay. So sarcomere basically is a contractile unit that exists in skeletal muscle and cardiac muscle. Really, that's all that you need to know as far as this class is concerned. So contraction occurs. Thick filament, the myosin filament or thick filament, they're thick, right? So they're pretty stiff. Microfilament, not so thick. They're thin filaments, and what happens when something is thin, they kind of flop like this. But for contraction, you need the microfilament to be completely stiff, right? So the stiffness of the microfilament or thin filament of sarcomeres is provided by intermediate filament. So along parallel to each of these microfilaments, you would have a Desmond filament. So the intermediate filaments that lies parallel to each microfilament is made up of Desmond, which is the monomer, the particular monomer. So again, this is cell specific. Not all cell would have Desmond as a monomer for intermediate filaments. So the intermediate filaments again provide stiffness for the microfilament. Okay. It's almost like if you want an analogy, if you plant a tree, a very skinny, thin tree, what do you need? You plant a post right next to it and tie the thin tree next to the post, right? So the post represents intermediate filaments. Okay, so they're apolar and have no motors. So I think that should be all of the very brief introduction to all three types in animal cell. There are only about 3 slides on prokaryotic cell, and we're very excited to talk about this. Is because even in microbiology, I don't think that you learn this in microbiology. So your career here, probably this is the only time you hear about cytoskeleton in bacteria. So there are only a few things that you need to know. Okay? We're not going to go into too much details outside of these 3 or 4 slides. Okay? So bacteria also have cytoskeleton. And as I mentioned before, the differences between a homolog and an analog is the fact that a homolog refers more to which one is which? refers more to, I think I said it opposite earlier, refers more to a common structure, okay? And an analog refers to more in terms of the common function. People in ecology, in Bio 1, you probably learned this, that for instance, the wings for butterflies and birds, they're analogs. They both are used for flying, but the structure are different. Right? The structure to a butterfly wings is completely different from how a bird wing is formed. Okay? So even though they have the same function, they have different structures. So that's analog. Okay? Versus, I guess, a bird and a bat wing would have similar structure, so that would be homologs. But if you have the same structure, chances are you will have similar function as well. Okay? And it really boils down to origin. How did the structure and function originate? Okay? So anyway, won't go into more detail than that. I'm not going to pretend I know much about all of that, but in terms of microtubules and microfilaments, and using the terms that we already determined, homolog versus analog, there is the homolog for G-actin. Okay, so structurally, the monomer that looks like G-actin would get assembled, but has a similar function to microtubule. Explain it in a different way. So microtubules, one of the functions of microtubules is during cell division is to allow to move the chromosome around, right? So in bacteria, you also have a cytoskeleton that can move the chromosome around. In this case, just a single circular DNA. Does that make sense? Right? So that is a similar function as microtubule. However, when you look at the actual structure of the monomer, the structure of the monomer looks more like G-actin than tubulin. So par-M is a monomer that looks more like G-actin, so it's homologous to G-actin. But when par-M is put together to assemble, right, that is assembled together, so the par-M filament would act more like microtubule. Versus FITZ, that's how that's pronounced, it's FTSZ, is FITZ looks more like tubulin, but it performs the same function as actin filaments during cytokinesis, where the contractile ring is formed. Okay, so basically, how you remember this is that the equivalent activity when you look at cell division, the same activity that occurs in eukaryotic cell also occur in prokaryotic cell in terms of the chromosome has to be moved around, as well as the cell has to be pinched into two, right? The chromosome that has to be moved around in eukaryotic cell is done through assembly and disassembly of the microtubule. And the pinching of the cell is through the activity of microfilament. Okay? So in the prokaryotic cell, you will have the same function, right? You will have the same function where you do have movement of the chromosome around. So that's similar to the function of microtubule. You also have the pinching off of the cell, similar to the function of microfilament. However, the monomers that make up these polymer, right, are are homologous to the opposite monomer than in the eukaryotic cell. Does that make sense? If you remember that, just remember opposite. Just remember opposite. That's how you remember this. That's how I remember it, by just thinking opposite. Okay. So PAR-M is an analog, is a homolog of actin, a G-actin, and PIT-Z is a homolog of tubulin. However, PAR-M filament is an analog of microtubule function, and PIT-Z filament is an analog of microfilament function. Again, this is, unfortunately, this is just something that you just need to memorize, and I don't have any rhyme or reason for it. Okay, so those are the only two slides. We used to have more. We used to have Flagella, the different. We might actually have it later on. We will actually later on when we talk about movement, cell movement. We'll compare flagella in a flagellum of eukaryotic cell versus a flagellum of a prokaryotic cell as well. So we'll just hold off for now. Okay, so let's look at cytoskeleton associated proteins, and all three cytoskeletons actually have associated proteins. So you have microfilament associated protein, you have microtubule associated protein, and you have intermediate filament associated protein. In terms of microfilament and microtubule associated protein, one of the major associated protein is a molecular motor, or motor proteins, right? For microfilament, the motor protein is myosin. So again, for microfilament, the motor protein is myosin. For microtubule, there are two motor proteins, kinesin and dynein. Okay? And since I'm a Texan, I pronounce it kinesin. Some people pronounce it kinesin. I don't care, right? Just know that that's how you spell it. So the difference between the two is that combined, kinesin and dynein performs the same function as myosin. So the function that is performed by myosin is split into kinesin and dynein. And later on, when we talk about the actual movement, intracellular movement, you'll see why it is that you only need one myosin, right? And then you need both kinesin and dynein. Why the activities have to be split. So myosin basically, and this could represent a myosin or any of the molecular motor. They all look similar, right? There is a kind of a tail here and 2 feet, or a head, I guess, and 2 feet, or a tail and 2 heads. However you look at it, right? This is the part that actually walks. The 2 feet here, or some people call them the 2 heads, actually move this way, so that they can. That's why they're molecular motors. They move this way to move materials. Okay. Myosin can move along the microfilaments either toward the plus end or the minus end. So myosin can move back and forth along the microfilaments, carry materials back and forth. Whereas for microtubules, kinesin can only move materials toward the plus end, and dynein can only move materials toward the minus end. So again, the same function that myosin does, which is move both ways. Kinesin and dynein, one moves one way, one moves the other way. Kinesin moves material toward the plus end of the microtubule. Dynein moves toward the minus end of the microtubule. And I know the term MTOC continues to show up, and I promise you I will explain what that is later. Are we okay with that? With that explanation? The only thing, this looks really complex, and it is fairly complex, but the only thing I need you to know about this is the fact that signaling has to do with everything. So how do you know? Take a look at myosin. Myosin can move either toward the plus end or the minus end. How does this myosin know to move toward the plus end or the minus end? Signals. signal from outside. Signal from outside telling the myosin that it needs to move in one direction or the other. Right? That's all that this slide represents. Is the fact that you may have myosin, and myosin can move either way, but there's specificity to the movement of myosin. The myosin is bound to a certain molecule or structure, right? And it is told to either move toward the plus end or minus end very specifically by outside signal. So myosin, without that signal, the myosin doesn't do anything, right? It has to receive that signal. So that's what this slide represents. Again, just another pretty picture depicting a molecular motor, where you have a tail here and two heads. I like to think of these as feet, just because they walk with your head, but people call these the myosin heads. So don't need to memorize this or anything like that. This is just a pretty picture for you to look at. Okay? Are you fairly comfortable? You don't have to be an expert. I'm certainly not an expert, but are you fairly comfortable with what each of the cytoskeleton, what it is, how assembly occurs roughly, and what are the functions, and the fact that what type of cells they're found in, right? Animal cell versus plant versus prokaryotic cell. Now what we're going to do is take everything that I talked about, okay, and then look at each in detail, and then at the same time, right? At the same time, apply to a function. So we're going to talk about assembly and disassembly of each of these cytoskeleton, right? But when we talk about assembly and disassembly, what is that really? That's really translational modification, right? And when we talk about translational modification, we have to talk about regulation of certain structure, certain function. Okay? So we're going to cram the regulation of assembly and disassembly in explaining function. Okay? So again, that's why I introduced all this ahead of time. So now when I talk about it, I can just say G-actin. G-actin bound to ATP without having to explain what that is. Okay? So regulation of cell function via control of that cytoskeleton. Let's do the same thing again. Look at microfilament first, and then microtubule, and then intermediate filaments. Okay? So this is a very dense, extremely dense slide. You will need to know every single last bit of this slide, by the way. So what I'm going to do is spend the rest of the time now, and definitely some, and we're not even going to finish, but at least the rest of the time now to explain the regulation of assembly and disassembly of microfilaments, and how then the assembly and disassembly relates to certain function. And again, I'm going to pick all of this apart. Okay? So the very first thing that we have to talk about, I don't remember what the next slide is. Okay, that's fine. The very next part is if you have a cell, right? And the cell needs to undergo assembly of microfilament. Okay? Assembly of microfilament occurs by the process of polymerization. So in terms of assembly and polymerization are synonymous. So there's only one. So polymerization is a mechanism. Assembly is a process. Does that make sense? Right? So assembly is a process. And the process of assembly is done through polymerization. Whereas disassembly, breakdown of microfilament, right? Disassembly is a process, but there are two different mechanisms that can be done. Through either depolymerization or severing. I will make that distinction later on, but I just want to show you to define the terms, and so that you can understand why is it that I use the term assembly and polymerization synonymously, but when it comes to disassembly, I very specifically talk about depolymerization versus severing. Okay? Because again, assembly and disassembly are just the process, the overall process. polymerization, depolymerization, and severing are the mechanism, right, that have to be performed. Okay. So for assembly or for polymerization, to start out, right, or you can think of that as the initiation of microfilament assembly, right? The initiation of microassembly is referred to as nucleation. Is that a term in here somewhere? Nucleated. So nucleation is the same thing as the initiation of microfilament assembly. Okay, I'm going to go through the next 20 or 30 slides as slowly as you want me. So I want you to understand all of this. Okay. However, so we can say the term nucleation. So nucleation of microfilament can be done in two different ways. Okay. Nucleation of microfilament can be done from scratch. So you don't have any microfilament, and then to do it from scratch, then nucleation is catalyzed by or is aided by a structure called foramen. So foramen again is involved in the nucleation of microfilaments basically from scratch, basically. Whereas ARP stands for actin-related protein. ARP aids or help in the nucleation of microfilaments on an existing microfilament. So if you have an existing microfilament like this, then nucleation can occur going in 70 degrees or 90 degrees from this. So think of the gel, going back, think of the gel-like structure, right, of bundling. Then in order to have the gel-like structure, then ARP is used, not foramen. Right? So either, not both, but either foramen or ARP may help or aid or catalyze the nucleation of microfilament. Okay? And then we'll look at all of these later, but that's one thing at a time, nucleation. So looking at foramen first. If foramen is represented by these green structures here. Okay? So what happens is this. Actin, and we see this in vitro. We see this in a test tube. And I mentioned this earlier. If you take a test tube and with a solution of some kind, water, and you add G-actin that have ATP bound, guess what? These G-actin will aggregate. Now, will they form a long microfilament? No, but they will aggregate. A few of them will aggregate. So we'll say that a few of them will aggregate spontaneously. Okay? And if you have an aggregation of a few, doesn't matter what how many a few are, but if you have an aggregation of a few actin filaments, then foramen can actually bind to that aggregate of microfilament. So far, so good? Now, it's a little bit more complex. Later on when we talk about a microfilament, right, and then severing occurs where you cut right in the middle, and then in order for the cell to tell that microfilament to grow again, foramen will also have to bind. So foramen helps or aid in the nucleation of microfilaments from an aggregation of G-actin. Okay. Let's see. Whereas ARP, so if you have a pre-existing microfilament, and we're not talking about the end here, we're just talking about a pre-existing microfilament, then right in the middle of that microfilament, an ARP structure can just plop down and then carry out nucleation. So then you get this gel-like structure. Is that, you see the difference between foramen and ARP? Both of them will carry out nucleation of microfilament, but they do it in different ways, and there are different needs for them. Okay? Yes? No? So that's one level of regulation. Yes, sir?
So the ARP just splits out from the center.
This ARP just, think of a potted plant. Right? If you want to plant something, you can put it, you can think of a tree, and then you can think of the fact that you put a potted plant and glue it onto the side of a tree and it grows that way. So the pot represent ARP, and then the tree can grow that way, right? That's a terrible analogy, I know, but visually you can see that, I think.
Are there like specific sites where you can do it?
And that's the point, is that anywhere along the microfilament, that's a good question. So anywhere along the microfilament, how does the cell know where along the microfilament? There are regulatory proteins that determines that. So is there a specific site? Yes, it's site specific. We just don't need to know that, but that's a good question. Yes, ma'am.
So the ARP is related to gel formation or gel structure?
Right. Yes. That's the point. Yes and no, actually. Yes and no. So if you were to, let's say on this, right? Let's say this is this is the cell cortex, and this is the plasma membrane, and then you want a microvilli up here. Then actually on the pre-existing microfilament, you may have multiple ARP that can carry out nucleation of multiple microfilaments, and then they are tightly bundled. So
Yeah.
you see that. So I understand your question, but it's a little more complex because of what I just said. Right? But you get it. Okay, good. Okay. So let me 5 minutes. That's enough time. The next thing that we have to be concerned with is part B. So part A, we're good. We're good with part A, right? Okay. Part B. Thymosin and profilin. Thymosin and profilin are regulatory proteins. Okay? To determine the speed, the rate of assembly and disassembly. So as I mentioned, this must be about the third or fourth time I mentioned this, that if you have G-actin around and if the G-actin had ATP bound to it, chances are that G-actin, ATP-bound G-actin can be used for polymerization, and so microfilament assembly can actually occur spontaneously at a low rate. But to regulate that rate, to increase or decrease, to mainly increase the assembly rate or mainly increase the disassembly rate, then there are two regulatory proteins that work against each other. Okay, one is thymosin, and one is profilin. So a G-actin, an ATP-bound G-actin can be used for polymerization and will be used for polymerization. However, if that G-actin were, that ATP-bound G-actin were also bound by a molecule of profilin, then the rate of microfilament assembly is greatly increased. Profilin is not an enzyme, by the way, so don't say that profilin is an enzyme that increases the rate. It's just that profilin is just a binding protein, but profilin then, by binding to the ATP-bound G-actin, actually, what does it do? Immediately causes a conformational change, which then can increase the affinity of the G-actin to the existing, on the plus end, to the existing microfilament, right? So again, profilin can bind to an ATP-bound G-actin to increase the rate of assembly. Profilin will never ever bind to an ADP-bound G-actin, by the way. That's the first thing. The next thing about this is an ATP-bound G-actin would have the same affinity for both profilin and thymosin. So either profilin and thymosin can bind. So if thymosin bind to the ATP bound G-actin, right? So we're talking about the ATP bound G-actin. If thymosin binds to the ATP bound G-actin, then that G-actin will not be used for polymerization. So thymosin then either slow down or even inhibit polymerization outright. So at that level of regulation, so we talked about the regulation of nucleation, right? Which ARP or formin must be present for nucleation to even start to begin with. And then when nucleation occurs, then the next level of regulation is whether or not thymosin and profilin are present to increase or to regulate the rate of assembly. Not to regulate assembly, but just to regulate the rate of assembly. So that's the next level, right? The third level is, since ATP-bound G-actin can bind to both profilin and thymosin, how do you regulate which one binds to the ATP-bound G-actin? Regulating the amount of thymosin and profilin. If you increase the rate of thymosin, my left hand is thymosin, if you increase the amount of thymosin, right, versus profilin, then that signals the cell to either inhibit or slow down assembly. If you increase the rate of profilin relative, again, everything is relative, you increase the amount of profilin relative to thymosin, then that signals the cell to increase the rate of assembly. Does that make sense? So that's the third level. So nucleation is one level of regulation. The use of thymosin and profilin is the next level. And then the third level is the relative amount of thymosin versus profilin. Okay? There's more regulation. But I ran out of time. Let me answer this question and then we'll just go home for the week. Yes, ma'am. Have a great weekend, everybody.